Welcome back, folks, to the next segment. In this segment, we're going to look at the role of DNA and a very important process of DNA replication. So let's try and understand what is the role of DNA in a cell. So when we try and understand the role of DNA, it's important for us to look at the different roles that DNA plays. Firstly, we're going to look at the role of DNA in the process of replication, which is fundamental to our lesson today. We're also going to spend some time looking at encoding information. So what does that mean for a cell? We also know that DNA has an important impact on mutations and recombinations of genetic information. And what we also need to spend some time looking at is gene expression. Why are certain genes switched on, and in which cells are they switched on? So, if we try and understand replication as an overview of the role of DNA, we know that cells divide, and in order for cells to divide, there needs to be some process of organization. So when a cell divides, the chromosomes containing DNA strands must replicate. And another word for replicate is copy. So it's important that the DNA of a cell has to be copied or make copies of themselves, so that when we produce two cells or daughter cells, that these two cells receive a full set of genetic information. So the rationale would be that if we've got a cell that divides to produce two new cells, that these cells must have an exact copy of that genetic information. And so it's important that that information is copied so that these daughter cells have that information. And so that copying process is replication of DNA. The second role of DNA is encoding information. And if you think about the word encoding, it comes from the word coding. So essentially, the sequence of bases in a gene determine the sequence of amino acids in proteins, which essentially is an important biochemical unit of the cell structure and function. So if we go back to, or we look ahead in terms of what happens next, we know that genes code for characteristics. And so this essentially means that in order for certain processes to happen, for example, protein synthesis, that information needs to be coded for by the DNA. So we're looking at how DNA plays an important role in coding for information that makes structural components like proteins, like enzymes. Mutations and recombination. DNA plays a role in the evolution of a species. Chromosomal DNA helix don't usually interact with each other. However, through the process of genetic recombination, segments of different chromosomes swap places with each other, creating new sequences of genetic material. So we refer to mutations as what drives changes in species. The fact that we have the genetic recombination when two alleles from parents, from a sperm and egg, come together. There is a combination of characteristics, and that talks to how the genes in that combination are expressed. So we refer to the recombination during sexual reproduction, creating combinations of characteristics that are different in the offspring. We also know that mutations can randomly occur, and those mutations are driven by the process of replication going wrong. Yes, something that goes wrong that gives an individual a mutation can be an advantage, could be a disadvantage. And essentially that is what drives variation in a population, and that variation leads to some being better suited than others. And so DNA is important in that process as well. We also know that there's an understanding that gene expression is unique in a cell. So we know that we've got different cells, and in different cells, certain characteristics or processes are stimulated. Those cells need to be switched on to produce a specific protein. For example, the cells in the pancreas will produce the hormones needed for regulation of blood sugar. There are certain cells that will produce certain enzymes in different parts of the body. And these we refer to as gene expression. So let's understand that. So each cell contains a full complement of genes. So all our cells have the same number of genes. Yet, cells from the different tissues and organs look and behave very differently. As I mentioned, the reason is that some of the DNA of each cell is used to make proteins. Not all. So certain genes are switched on. So we say that DNA plays a role as a traffic cop for the types of proteins a cell will make. It does this through interactions with proteins in the cells caused only certain genes to be expressed and to express themselves. So we say that not all genes are expressed in every cell. Certain factors regulate the expression of genes in a cell. And so we've looked at the role of DNA. Now let's get to the process of understanding replication. So what is DNA replication? If we refer to the term replication, replication essentially means copying. So it's a process where the DNA in a cell makes a copy of its cells during cell division. Okay? Where does DNA replication occur? It's a very interesting question. DNA replication occurs in the cytoplasm of prokaryotes. Remember that your prokaryotes are cells that do not have a membrane-bound nucleus, and hence it happens in the cytoplasm. However, in eukaryotes, these are organisms that have a defined nucleus. We know that it happens in the nucleus. However, what is important is that regardless of where the DNA replication occurs, the basic process which we're going to look at is the same. The structure of DNA lends itself easily to the DNA replication. Essentially, when we look at the structure of DNA, which is a double-stranded molecule having those complementary base pairs with complementary nucleotides on either end, it's that structure that leads to the process of DNA replication being universal. Right. So this is an illustration of when in the cell cycle this process of DNA replication occurs. We know that a cell goes through different stages, and what is unique is that the process of DNA synthesis or replication occurs in a phase of the cell cycle called interphase. We have looked at cell division, where we spoke about cells growing and dividing during mitosis and in meiosis, but we also know that the cell undergoes a process of specialization and development, and that during the synthesis stage it will go through what we know as replication. And so that is in interphase that the cell undergoes replication. And so that's key for our understanding, is that in interphase the cell will undergo the process of cell division. And we see this in mitosis and meiosis, and it's important that the cell is able to copy the DNA so that the daughter cells at the end of cell division have an exact copy of the genetic information. What is the significance of replication? The purpose of DNA replication is to produce two identical copies of DNA. This is essential for cell division during the growth and the repair of damaged tissue. DNA replication also ensures that at the end of each new cell receives its own copy, an exact copy of DNA. So when cells repair and regenerate, it is important that these cells have the same genetic information as the original cell that they've formed from. And so that is why it's important that DNA replication produces identical copies of these cells. So let's understand the process of DNA replication. It's important that we identify what is needed. And so there's a series of needs for this process to occur. The first in most biochemical process is enzymes. And the enzymes we're going to look at is a group of enzymes, and we refer to them broadly speaking as DNA polymerase. The focus is important that we recognize that these are DNA polymerase enzymes. We also need the building blocks. If we were to make DNA, we need the building blocks of DNA. And so it's important that we have the building blocks called DNA nucleotides. When copying or replicating, we need a reference, and that reference strand to copy from is the DNA template. So we're going to need a template to copy from. So in this illustration, I've outlined the process of DNA replication in a series of different steps that are illustrated in numbers. I'm going to talk you through this process so that we can understand essentially what happens. I'm hoping that in this illustration that we can go through each process trying to understand exactly how the DNA molecule is able to produce a new copy. So the first process in DNA replication is that the DNA will unwind and unzip. So here we see the DNA molecule. It's going through a process of unwinding and unzipping. If you refer to a zip on a jacket in a jacket will allow for you to open the halves that are combined together. Similar to that, DNA is held together by hydrogen bonds. And these bonds need to be broken up. So we have an enzyme called helicase that breaks the weak hydrogen bonds, and here you can see that enzyme illustrated in this by this arrow, which is an enzyme that breaks the hydrogen bonds holding the complementary base pairs together. And this causes the separation of the two strands of the DNA into templates on either side. So we now see that the enzyme breaks open the strands, causing a separation. What happens next is that these exposed strands that we see that have separated now act as a template. A template is essentially something we copy from. So if you take a worksheet to a photocopying machine, and if you place that over, the photocopy uses that as a template to make more copies. So we've got two templates that are now serving as a reference strand for us to copy new DNA or copy complementary DNA. So each strand acts as a template for the formation of new complementary DNA, forming a set of two DNA strands. So here we see on these further down, we see that this is the template on the one strand, and here we have the template on the other strand. And using the enzyme, and the enzyme in this case is DNA polymerase, that enzyme brings in the free complementary building blocks called nucleotides. So these templates are now exposed. We find that the nucleotides that are floating in the cytoplasm, the nucleoplasm, are brought in to base pair with their complementary strands on the DNA template. So the free nucleotides will move and they pair up with the exposed bases on the single strands. So here we've got these exposed bases, and so these free nucleotides will move in and they will pair up, which I've illustrated further down here. And this happens through an important set of enzymes called DNA polymerases. And this enzyme is able to match the bases, knowing that we've got adenine and thymine base pairing together, and guanine and cytosine always base pairing. So using that enzyme, you'll find that these templates will then serve as a template for the combination of a complementary strand. As this process occurs, the enzyme polymerase continues to read these strands, and it will help to base pair in the following order. So you'll know that a thymine will base pair with the cytosine, and likewise an adenine base pairs with the guanine. And as this process continues, polymerase continues to unzip the DNA, brings in more complementary free nucleotides to eventually form two new identical molecules of DNA, both of which are double-stranded. Each one will eventually twist to form a helical structure. So a simple illustration of the process would be that you've got a DNA molecule that then needs to unzip, and as it unzips, you're going to find that in the nucleoplasm, these templates now, which I'm going to enlarge in, okay, have on them the bases that are exposed. So if you have an adenine here, you've got a thymine there. If you've got a guanine here, you've got a cytosine there. And all of this is happening through the enzyme which is surrounding this, which is I've indicated as DNA polymerase. The DNA polymerase will help to bring in the complementary bases. And in this case, you've got adenine, thymine, guanine, and cytosine floating in the nucleoplasm that come in and base pair with their complementary templates to eventually form their complementary strands on either end. as illustrated, you will get a strand. I'm going to go back to my red pen, okay, that now forms on each, forming 2 new strands that have double-stranded complementary DNA constituting them. So if we were to wrap this up in a few steps, the steps are as follows. The DNA unwinds, unzips. An enzyme called helicase helps to break the hydrogen bonds, as I've indicated in here. So this is the enzyme that's going to break them up. And this causes the 2 strands to separate, creating what we refer to as DNA templates. Okay? The second process would be that these templates form, and each strand acts as a template so that new complementary bases can come in and form. So here we're seeing that the new complementary bases are coming in based on the ATGC base pairing, that they start forming a complementary strand on the template. The next step would be the pairing up of the nucleotides, and we see here that these nucleotides start pairing up with that ATGC rule that we know and we've just illustrated, and they start eventually doing this with the help of an enzyme called DNA polymerase, and they start pairing up together with the free nucleotides floating in the nucleoplasm, and using that, the complementary base pairing occurs. And we finally see that once these base pairs are joined together with the combination of A and T combining, we look at finally the helix forming, which we see here, and the DNA starts reconfigurating into a double-stranded helical twisted molecule. And guys, that is essentially a wrap of how DNA replication takes place. So we've looked at the process, we've looked at the stages in which it occurs, and fundamentally important is the process of how it occurs is for you to be able to put down into a flow diagram and list the important processes of how the templates form, the enzymes bring in the free nucleotides to base pair with the complementary strands, to eventually form 2 new strands that are identical to each other, thus forming copies of the original DNA. Guys, you've done well. Let's take a little break, and when we get back, we will look at how the process of replication is important in terms of the cell division, and we'll try some application of this to some questions. Short break, and see you in a little while.